Okay, good. So I'm Steve, I'm the CEO. I'm going to tell you a little bit about the story behind Xanamem, now called emestedastat. It's been given this new INN name with a unique suffix, stedastat, recognizing that this is the very first 11-beta HSD1 enzyme inhibitor ever that has got to the stage of receiving an INN name. Because this molecule is unique, gets into the brain. To our knowledge, no other inhibitor of its type has ever been able to test the cortisol hypothesis in the brain that a molecule such as ours can actually get in there, inhibit cortisol production, lower tissue cortisol in essential areas of the brain to reduce toxicity, progression in dementia, and as we showed this year in a phase 2A or last year, improve symptoms of depression in major depressive disorder. So we are a public company, so we'll be making forward-looking statements. And just very briefly, a novel molecule, a very well-behaved small molecule, positive clinical data in 2 clinical indications. Dana will explain this. Excellent patent protection, and of course we have data exclusivity protection in addition to patent protection with a new chemical entity and a unique species. And we have very large and important clinical opportunities in Alzheimer's, related dementias, and major depressive disorder, and potentially other psychiatric conditions. We have a good-looking board and management team. We are a semi-virtual company based on the Australian Stock Exchange in Sydney, but with global operations. Many of our key employees and our senior team are based here in the United States. And then lastly, as an opening comment, the new depression data showing clinically and at times statistically significant improvements in the MADRS depression score, and also the measure of patient reported outcomes of severity, really validate the program in a number of different ways. It shows that the 10 milligram dose we are using, while we had already proved it gets into the brain, it shows that it's actually doing something important clinically. Previously, we showed activity in patients with Alzheimer's and elevated p-tau. Now, in a completely separate indication, we're showing that inhibiting tissue cortisol in key areas of the brain has a major effect on symptoms. And that really validates everything we're doing in major depressive disorder, but really also reads through to Alzheimer's disease as well. So we have a pipeline that's focused on Alzheimer's and major depressive disorder. We also do have an open IND for Fragile X, where cortisol has a good scientific rationale, but clearly there are other indications that could be pursued in the near future. And with that, I'm going to hand over to Dana, who will spend much more time on the science.
So Steve has already made some comments outlining some of the properties of the molecule. I want to basically emphasize that this is a mechanism to decrease brain cortisol without affecting circulating cortisol. Okay? Isoform 1 of this enzyme exists in brain, liver, and adipose. And so when you give this, it's not the enzyme that makes cortisol in the adrenal gland. So that allows you to selectively decrease, largely, brain cortisol levels without having systemic side effects. And cortisol has been invoked in depression, in Alzheimer's disease. It's neurotoxic. There's a vast literature on this that's more than 20 years old. And because this is the first one of this class to actually get into brain, show that we can actually inhibit the enzyme in brain, we think this is the first time that this hypothesis can be rigorously tested. So there have been over 400 or approximately 400 patients treated with this drug. It's safe and well tolerated. There have not been any serious adverse events linked to the treatment with this molecule. It's brain penetrant. I'll show you just a snapshot of some of that data. And we have 2 phase 2 trials which show that it has clinical activity. In one phase 2 trial, it's a 3-month trial in patients with AD and high p-tau 181. It slows the CDR sum of box progression. That's a pilot study, which then led to the design of the phase 2B study that's now being conducted in 220 patients in Australia and the United States to look at the CDR sum of boxes in patients with clinical Alzheimer's disease and elevated p-tau 181. Those patients are going to progress over the observation period. And then separately, This year, we've read out a phase 2 trial in approximately 180 patients, showing 170 patients, showing that it actually has antidepressant effects. And I'll outline some of those data to you. Oops, got to go the other direction. No. Oops, sorry. Oh, big ones, yep, sorry. When you rely on a neurologist to do something as difficult as advancing it, it's very challenging. So what we're doing is reducing, as I mentioned, cortisol production in the brain. And when you think about this in a sort of a simplistic way, it's reducing the stress response in the brain. And as I mentioned, Bob Sapolsky and a variety of investigators have shown for more than 30 years, this could actually be toxic to neurons, hippocampal neurons, other neuronal populations in the brain. And the stress response in the brain cascades and produces deleterious effects over both the short-term and longer-term impact on brain function. Now, this is the study that was published this last year showing that this drug actually gets into the brain and binds the target. On the left is a probe to the enzyme, 11-beta-HSD1 isoform 1. As you can see, that's expressed in the neocortex widely. And the three right panels are the same probe plus 5, 10, and 20 milligrams of emestedastat. And what you can see here is it blocks the binding. So that shows the drug gets into the brain, binds the target, and blocks the effect. Now, in Alzheimer's disease, I don't have time here today to go into all the background epidemiologic data, but cortisol levels are elevated in CSF and in the brain of patients with Alzheimer's disease. As I mentioned, there's a vast literature that shows that chronic cortisol administration or corticosteroid administration can cause hippocampal atrophy and neuronal cell death. Elevated cortisols are associated with clinical progression in patients with AD. And in a variety of preclinical animal models, blocking this enzyme blocks the effective progression in those preclinical models. This drug is safe and effective. It's oral. It's a once-a-day therapy. It has low risk of interaction with other drugs. And so this could be added onto present therapy. I do not need to tell this audience that we haven't solved the Alzheimer's disease challenge yet. We finally have some drugs that have some benefit, but clearly those are not going to be the full answer to treating this disease. Here's one of the drugs. This is the monoclonal antibody. And shown here, what one can see is that the natural history of the disease, there's progression, and the CDR sum of boxes over 18 months. This is the graphically shown is the type of effect size you get when you totally clear amyloid with an antibody. So both lecanemab and donanemab clear amyloid with antibody, but this is the magnitude of the effect. Now, this difference, actually from the literature, is not clinically significant. It's statistically significant, a 1,000-patient study, yes. But I think if you think, the way I think about it as a neurologist, the area between the curve is the treatment effect. This is the unmet need, the area between these two curves. And so it's pretty clear that this chronic disease, like most other chronic diseases in medicine, will actually require combination therapy. So the antibodies alone are not going to actually completely cure the problem or solve the problem. In the initial study that Actinogen did, patients were enrolled who had a clinical diagnosis of AD with no biomarker determination. Now we know from the literature that if you do that you're actually wrong about 40% of the time. Many of these patients, particularly early in the disease, will just fluctuate. They don't progress. So what we did was, in a blinded way, prospectively, we went back and assayed these patients' plasma for p-tau181. We took all the patients that we had plasma on, looked at p-tau181, and we looked at patients that had low levels and high levels of p-tau181, and we looked at the treatment effect with emestedastat versus placebo in those patients. And what we found is that the patients with placebo had a significant progression even over 3 months because they had elevated p-tau181. And the patients on emestedastat had a blunting of this. Now, this is a modest sample size. This is a pilot study, but you can see the Cohen's d here is about 0.4. This didn't reach statistical significance, but that's only because of the modest size of the study. So this study then informed the next study we are doing, a larger study, a phase 2b study in patients with clinical diagnosis of AD and elevated p-tau181. That's the study here. And so this is 10 milligrams versus placebo, 9 months, qualifying the patients with a clinical diagnosis of AD, elevated p-tau181. The primary endpoint of this study will be the CDR sum of boxes. The secondary endpoints will be cognition measures and ADL measures. And this trial is underway. It was initiated first in Australia. It's now being opened in the United States. We've just opened about a dozen sites in the United States. This trial is being run as a phase 2b pivotal trial with all the bells and whistles. It has an independent data monitoring committee. There'll be an interim analysis. It'll be using standard state-of-the-art industry and FDA-accepted phase 3 statistical methodology, and it's being conducted with a commercial tablet formulation, the 10 milligram form. The interim analysis will be, we expect to have that data later this year, and then the final test results next year. On to depression briefly. So we all know in this room that there's a significant unmet need in depression. Most patients with depression, even those that are adequately treated, go through 2 or 3 different medications. Many patients don't take their antidepressant drugs because they have side effects that are not tolerable to patients. There's a very good, strong scientific rationale for the cortisol hypothesis in patients with major depressive disorder. I've listed some of the aspects of this here. You can block cortisol receptor activity with mifepristone, and that's actually had antidepressant effects. However, that drug is not tolerable long-term due to the side effects. And so what we really need here to access this pathway and produce a benefit is a drug that gets into the brain, blocks the production of cortisol, yet is tolerated systemically. That is actually emestedastat. And so what we did was a fairly robust phase 2A type trial to determine, does this drug have antidepressant effects? So that trial was 10 milligrams versus a placebo. It was a 6-week trial. Antidepressant trials are usually 6 to 8 to 10 weeks in length. The primary endpoint at this point, when we did this trial, we did not know whether emestedastat would actually have an antidepressant effect. We knew it had pro-cognitive effects. So we stated the cog-state cognition test battery as a primary endpoint in this trial. But the real clinical endpoints are the MADRS and the patient-reported outcomes, as you know. These are the types of scales that the FDA will want for a depression drug to approve a depression drug. Who did we enroll? The mean age was approximately 50. Predominantly, or majority female is typical. Now, we qualified patients for residual depression with a HAM-D, but we used a separate scale to measure clinical benefit. This is always something one should do in clinical trials if possible. Use a separate qualification metric than what you're using as an endpoint. So they had significant residual depression. Importantly, this group of patients, about 80% of them were on background antidepressants, yet they still had significant depression. This is not a full treatment-resistant population. This is not a ketamine candidate population of patients, but it's just before that. These are patients that, in spite of being well treated with an accepted and approved antidepressant, have significant residual depression. Okay? And they also had a cognitive deficit. What we showed over time, first on the MADRS, is now this mechanism is very different than modulating neurotransmitters. It appears that it takes longer to see a clinical benefit, but you start to see a clinical benefit out here at 6 weeks, and that actually increases over time. And if you look at the patient-reported benefit, you actually see separation earlier with an effect through 6 and 10 weeks. And when you look at patients that are actually on SSRIs, we enrolled patients on SNRIs and SSRIs, which is, as you know, SSRIs, a predominant class of drugs used to treat these patients. We actually had a bigger effect. So by 10 weeks, you see a Cohen's d of 0.64 and a p-value of 0.03. And this is the SSRI patient population for the PGIS. We also did a responder analysis, and we showed that using two well-accepted definitions of remission, more than a 50% reduction in the MADRS and percent with 10 points reduction in MADRS. It was positive on both of those. And this is not an eye chart. This is what I want you to do is to get the gestalt of this. What I'm showing you is with these endpoints, a MADRS with a Cohen's d greater than 3, statistically significant, or a patient response Cohen's d greater than 3 and statistically significant. At the numbers represent the time points, and green favors treatment versus placebo, and red favors placebo. And what you can see from that very clearly is that the majority of the effects favored the treatment versus placebo in the study. And it has an excellent safety profile. Again, no serious adverse events. And so briefly, this study we think is going to inform the design of a phase 2b trial in depression. We're taking these data to the FDA and other regulators for discussion on the first half of this year. This study was done in a heavily pretreated population. The next study will be done in patients that aren't on background therapy. The subset of patients in the first trial that had no background therapy had a more robust response. And so in conclusion, we think these are two separate clinical programs where the drug has shown both biologic proof of concept and clinical proof of concept. And this has informed the ongoing phase 2b and will inform the next study in depression. Thank you.